Hello everyone, it's a pleasure to meet you for this second webinar organized by the Data and Partnership Department of UNICANCER. This webinar focuses on the topic of methodological challenges for use cases with real-world data and will be hosted by two prestigious speakers, Louise Baschet, co-founder and head of Advanced Methods Department at Horiana, and Thomas Filleron, head of Biostastique and head data science at Oncopole Claudius Regaud. There will be a 40 minutes presentation followed by 20 minutes of question. You can answer your question through the chat. Thank you for your participation.
Okay, brilliant. So thank you, Daniela, for the introduction. So just before Louise Baschet and Thomas Filleron are going to present so this new methodological challenges for use case with real-world data, a bit of context concerning UNICANCER real-world data program. So we from UNICANCER has built several programs to retrieve real-world data. We have a retrospective cohort like Oncodata Hub in breast cancer and lung cancer. ESME cohort as well. In metastatic breast cancer, advanced and metastatic lung cancer, and which has been referenced by HAS, so French Health Technology Assessment Body. Prospective cohort in early breast cancer and early lung cancer, CANTO cohort, and the fourth program, which is a WISHER, which is a digital approach to retrieve data, especially for patient-reported outcome. And it permits, in a nutshell, to have this map of the program that we have, because today the topics that will be addressed by Louise and Thomas are concerning use case for health technology assessment body, so regarding what we can do with real-world data concerning discussion on dossier which are submitted to health technology assessment body. The topic of external control arm, which means to the use of this methodology in order to fine-tune a clinical trial, but as well to be compliant with a process of assessment of health technological assessment body, and the limit on benefits for this method. And what we see at UNICANCER, it's in the continuum of development of an asset, either if it's like a pharmaceutical compound or a medical device. We have the use of real data for early asset strategy, so it means to pinpoint what are the targets for unmet medical needs in a large cohort to have this kind of information, especially for toxicity resistance mechanism. So really early in the process of research. It permits as well to fine-tune business cases, so assumption for a specific indication or a biomarker subpopulation, and for clinical development, the use of a synthetic control arm in the clinical trial design, and for regulatory, it's much more to address questions about the course of disease, treatment pattern, and real-world outcomes that can be assessed in this large real-world data program. And in the end of the maturity of the development of a compound or a new strategy in pharmaceutical development, as well as economics and outcome research and market access, and as well as the reuse of synthetic control arm, as well and the cost effectiveness analysis, which permits to have much more informative data in order to produce produce a dossier addressed to a health technology assessment body. So it's a real pleasure to have you. You're very numerous today to attend to the meeting. So it's a great pleasure and honor to welcome you and the speaker for Louise and Thomas. So the floor is yours, and very happy to share your expertise, and do not hesitate to prepare your question on this very interesting topic. So the floor is yours.
Thank you. Thank you very much, Goodman and Daniela, for the presentation. I will share the screen. Can you see it?
Perfect for me.
Yes.
Yeah, perfect. So thank you very much. I am Louise Baschet. I am a biostatistician for 18 years now in clinical research. And I am pleased here today to speak about real-world data in oncology uses and new perspectives, with a focus on HTA bodies assessment and also synthetic control arm.
Hello, my name is Thomas Filleron. I'm the head of the Biostatistics and Health Data Science unit in the Oncopole Claudius Regaud, and I'm also an associated researcher in Oncostat team Villejuif. In the first time, we would like to thank UNICANCER RWD partnership who invite us to talk on real-world data in oncology use this and new perspectives. Real-world data and real-world evidence is actually in the rise. In a recent LinkedIn post, ESMO reveals that 50% of all submissions at last ESMO were real-world studies. We conduct a rapid pre-med research to identify publications containing real-world data or real-world evidence within the title or abstract. These terms start to appear in 2017. The number of publications has rapidly increased between 2020 and 2022. This was verified for both oncology and non-oncology publications. During last ESMO, a comprehensive mapping review of published real-world evidence focusing on targeted therapy was presented. The author obtained similar findings concerning the number of publication increase. However, they note that few manuscripts were published in high-impact factor journals. Last week, a guidance for reporting real-world evidence studies specifically for oncology was published by the European Society of Medical Oncology. This publication provides guidance for full article development. A comprehensive list of detailed key recommendations is proposed to improve real-world evidence publication. Real-world data can also provide a basis for synthetic control arms in single-arm trials where randomization in a multi-arm trial would be unfeasible or unethical. The topic of external control is in the rise, and the number of publications increase over time. Using regulatory case studies, multiple groups have examined methodological challenges for designing external control arms. They have suggested strategies to mitigate them.
Thank you very much. We can go back to history. Historically, the studies was mostly designed as one size fits all, and patients suffering from cancer are treated on the basis of their tumor sites of origin and histological subtype. Oncology practice relies on average population benefit decisions, often derived from randomized clinical trials on unselected patients that answer to selection criteria not specific to their type, but only on the tumor sites of origin. Indeed, randomized controlled trials are considered as the gold standard. Although no study is likely on its own to prove causality, randomization reduces bias and provides a rigorous tool to examine causal effect relationships between an intervention and outcome. But conducting a randomized clinical trial can be difficult and have several limitations. One drawback is that it is time and cost consuming, leading to long time before treatment access for the patient. It is strongly protocolized with limited representativeness participants that volunteer to participate might not be representative of the population being studied, and also limited external validity. It is restricted to sample size estimated to answer to the primary objective with mainly no long-term efficacy endpoints. It is designed to assess efficacy and safety for the included population without answering for each profile of patients' characteristics. And finally, it can be not feasible for several reasons.
The healthcare has undergone massive change with the development of molecular target agents and immune checkpoint inhibitors. Scientific breakthroughs and technological advancement have improved the understanding of disease pathogenesis. So the understanding of the cancer biology evolves, and the accessibility to tumor genomic sequencing technology increases. Consequently, genome-driven cancer treatment emerged as a promising strategy. This development allows the emergence of precision medicine. The aim of precision medicine is to provide the right treatments to the right patient at the right time. The ancestral paradigm of drug development in oncology is further challenged by the emergence of precision medicine. With the emergence of molecular target agents, the realities of the conventional screening approach in clinical drug development are sobering. Over the past 10 years, non-small cell lung cancer has become the archetype for precision cancer medicine. Molecular testing of lung adenocarcinoma for oncogenic driver mutation has become standard in pathology practice. Patients receiving a molecular match therapy showed a significantly higher survival probability compared to patients receiving conventional chemotherapy. In the pivotal phase 3 trial, which compared crizotinib to chemotherapy in advanced ALK-positive lung cancer, 4,967 patients were screened during 2 years. 588 patients presented lung cancer during ALK rearrangement. Finally, 347 patients were randomized from 105 sites in 21 countries. To date, various molecular target agents are under investigation in cancer for alteration less frequent than 5%. As example, ALK translocation 4%, RET translocation 2%, HRD mutation 1%. Also less frequent, some mutations could be relevant in patients. Biomarker-driven drug development represents a challenge when the prevalence of the biomarker is low. Consider a tissue-agnostic drug candidate in a subpopulation of patients. This subpopulation of patients was selected by a biomarker with a 2% incidence. If 100 patients are screened, 2 patients with mutations will be identified to finally include approximately 1 patient. Under this hypothesis, it's necessary to screen 14,300 patients to include 200 patients in a trial. In this setting, randomized comparative trial may not be a feasible option, and it's necessary to conduct single-arm study.
Thank you. What is the EMA point of view about these questions? 2 years ago, the European Medicines Agency published a position paper with this analysis, compared with drugs from the blockbuster area. Recently authorized drug and those expected in the future present an heterogeneous mix of chemicals, biologicals, cells, and gene therapies, a sizable fraction being for rare disease, and even individualized treatments or individualized combinations. The shift in nature of products entails secular trends for the definitions of drugs and target population, and for clinical use and evidence generation. In this paper, they discussed that the lessons learned from evidence generation from the 20th century medicines may have limited relevance for the 21st century medicines. They explain why the future is not about randomized clinical trials versus real-world evidence, but RCTs and RWE, not just for the assessment of safety, but also of effectiveness. This year, they analyzed the contribution of RWE in submitted dossiers. They characterized RWD and RWE presented by applicants to support claims on medicine's efficacy within initial marketing authorization application, MAAs, and extension of indication applications. RWD-RWE was included to support efficacy in 32 MAA and 14 EOLs. submitted between 2018 and 2019. Of those, RWD, RWE was considered supporting the regulatory decision in 10 applications. If we have a look at the FDA point of view about RWE, as described on its website, FDA has a long history of using what we currently call RWD and RWE to monitor and evaluate the post-market safety and approved drugs. RWE has also been used historically to support effectiveness, but on a more limited basis. Advances in the availability and analysis of RWD have increased the potential for generating robust RWE to support FDA regulatory decisions. As an example of the FDA's work on these topics, the guidance for industry for the use of RWD and RWE to support regulatory decision-making for drug and biological projects has been published last August by the CDER, CBER, and OCE.
Thanks, Louise. The terms real-world data and real-world evidence are being used inconsistently and sometimes interchangeably. Despite the increasing popularity of real-world data, there are a lot of different definitions. This imprecision has complicated efforts to assess the impact of such data and evidence. In a literature review by Macali et al. Suggests classifying the different definitions in 4 categories. Data collected outside of tightly controlled trials. Data collected without interference with treatment assignment or patient monitoring or selection of study populations. Data collected in a non-experimental setting. And finally, the last categories correspond to definitions that do not fit on the other 3 categories. Per the definition by the FDA, real-world data in the medical and healthcare fields are the data relating to patient health status or the delivery healthcare routinely collected from a variety of sources. There exist several potential sources of real-world data: patient registries, electronic health records, administrative claim, patient-reported outcome, observational studies, pragmatic clinical trials, early access program. If analyzed appropriately, such data can give rise to so-called real-world evidence. Per the definition by the FDA, real-world evidence is a clinical evidence about the usage and potential benefit or risk of a medical product derived from analysis of real-world data.
Thank you. So what for and how to use RWD? What for? When possible, it's allowed to leverage limitations of RCTs. It can produce evidence closer to reality, and it's aimed to permit a faster access for patients. And how? Hybrid trials methodology have been developed, combining the best parts of traditional randomized clinical trials and observational study designs to produce RWE that provides adequate scientific evidence for regulatory decision-making. So some hybrid design include randomization and use pragmatic outcomes, as Thomas just mentioned. Other designs use single-arm trials that are supplemented with external comparators derived from RWD, or leverage novel data collection approaches to capture long-term outcomes in real-world settings. And recently, emulated trial using RWD has been suggested to leverage observational studies.
To illustrate the different concepts evocated by Louise, suppose a hypothetical trial with 2 treatment arms, denoted A and B. Treatment A corresponds to the experimental treatment. In randomized comparative trials, patients are randomized between the 2 treatment modalities. With properly designed randomized clinical trials, all confounders will be balanced between the 2 groups. Randomization allows to balance both mutual and unmutual confounder. In a single-arm trial with external controls, patients are prospectively enrolled in a single-arm trial. Because there is no randomized comparator, all patients in the trial receive the experimental treatment. Externally controlled trials include a comparator group derived entirely from a source of secondary data. External controls could be selected from previously completed randomized clinical trials or reconstituted from real-world data. Most of the time, population differ in characteristics that may also affect the outcome. Ignoring those differences will lead to misleading inferences owing to confounding bias. To reduce confounding bias, statistical methods such as propensity score are used to build a control arm and balance observed disease characteristics and demographics. Finally, target trial emulation has been proposed as an observational method to answer comparative effectiveness questions. Patients are treated according to the local prescribing information and routine medical practice. Target trial emulation is a method in which investigators explicitly mimic an idealized clinical trials using observational techniques.
Thank you. We are going to do a small focus on external control arm. So what is it? Also called synthetic control arm, it's a control group that is derived from external sources, such as published data, previous clinical studies, or population-level databases, such as real-world historical data. What for? To conduct externally controlled trials in order to estimate a new treatment or health product effects and improve regulatory submissions. As an alternative to randomized clinical trial, it allows to accelerate clinical developments and then the health technology access to patients. When is it particularly relevant? In case of difficulties for recruitment, as in rare diseases or uncommon conditions, or if it's deemed unethical to randomize patients to no treatment or placebo group for life-threatening conditions. And how? To reduce the potential bias and increase confidence in the interpretability of study results, it is recommended to finalize the study protocol, including selection of the external control arm before initiating the single-arm externally controlled trial. Several steps are mandatory, and each presents its own challenges. The first is anticipation during the phase 2 or phase 3 design. Then the design to ensure rigorous methodology. Then the data selection and collection with sufficient quality. The quality of the data is very important for the analysis. Then to conduct the relevant analysis and to report it transparently with the good communications. Today, HTA bodies are working on guidelines. If we go back to the previous submissions, an interesting paper published 2 years ago has compared previous guidance documents from FDA on RWD, rare disease, serious conditions, and EMA through the International Conference on Harmonization, ICH, ITAN, about the choice of control group and related issues in clinical trials. HTA bodies estimated that use of external controls is acceptable under certain circumstances. And this paper also studied the use of external controls in FDA regulatory decision-making between 2000 and 2019, and 45 approvals were identified where FDA accepted external control data in their benefit-risk assessment. They did so for many reasons, including the rare nature of disease, ethical concerns regarding use of placebo or no treatment arm, and the seriousness of the condition, and the high unmet medical need. Retrospective natural history data, including retrospective reviews of patient records, was the most common source of external control. Other types of external control were baseline control, published data, and data from previous clinical studies. HTA bodies are working on these subjects with several recent publications. The draft guidance of the FDA on the consideration for the design and conduct of externally controlled trials for drug and biological products has been published in February this year. Also in February, the French HAS methodologists published a position paper on rapid access to innovative medicinal projects while ensuring relevant health technology assessment. And the second part of this paper focuses on external comparison. And the reflection paper on establishing efficacy based on single-arm trials submitted as pivotal evidence in the marketing authorization was under public consultation for by the EMA until the end of September.
Thanks, Louise. With the emergence of molecular target agents, single-arm trials with external control have gained popularity in oncology. More concretely, over the past 20 years, the FDA has approved 176 indications for novel agents exclusively on the basis of single-arm studies. When a randomized design is not feasible, incorporating external control data into the study design can be an effective way to expand for the interpretability of the results of an experimental treatment by introducing the ability to carry out a formal or informal comparative analysis. This paper provides an introduction to the concept of external control in oncology trials, followed by a review of relevant and current research on this topic. The paper also focuses on general consideration for designing a trial that may incorporate external control data. Case studies of the marketing application submitted to the FDA were discussed. Concerning future direction, authors also said, it's possible that formal analysis for comparative treatment effect between an investigational therapy and an external control arm would provide the primary evidence of efficacy to support a regulatory approval in oncology. This point is particularly true for disease type with rightly predictable natural history and the precisely measurable treatment effect.
We can have a look on several examples about indirect comparison. A recent example is the Libtayo submission. So last August, the HAS in French gave a favorable opinion for reimbursement in the subgroup of patients with metastatic or locally advanced cutaneous squamous cell carcinoma who are not candidates for curative surgery or curative radiotherapy and who are not eligible for chemotherapy. And this assessment was based on an indirect comparison called TOSCA, comparing indirectly data. The first arm is from patients treated with the treatment Libtayo between April and October, in 2019 under its early access cohort. And the second arm, the external control arm, was an historical cohort of patients treated in France in the same expert centers with systemic treatments used off-label before the treatment Libtayo became available between 5 years between the period of early access cohort. And the method to do that was an inverse probability weighting on confounding factors. And the confounding factors were selected by systematic literature review. And some variable couldn't be considered as ECOG, for instance. A second example is the Kymria CHMP from the EMA positive opinion for granting on marketing authorization on indicated for the treatment of adult patients with relapsed or refractory disease large B-cell lymphoma after 2 or more lines of systemic therapy. This assessment was based on indirect comparison of the single-arm trial called Juliet and a cohort, SCHOLAR-1, which combined several data sources like previous clinical trials or RWD. The method to do that was a matching adjusted indirect comparison called MAIC with the application of the same selection criteria, the weighting taking into account 3 variables, and after that, the comparison after the matching or the weighting. And we can also present a last example. It's comparison between the ESME cohort to 3 aggregated single-arm trial for entrectinib. And to do that, we use a matching adjusted indirect comparison. And because of several statistical challenges like missing data and small sample size, we needed to develop several methodological aspects to ensure transparency of this analysis.
Thank you, Louise. To minimize bias, external control studies should apply a rigorous process. A 3-step guidance was recently proposed by Jérôme Lambert and colleagues. The first step includes the definition of an estimate reflecting a clinical question. The second step relies on the adequate selection of external controls from previous randomized clinical trials or real-world data. The third step consists of choosing the statistical approach targeting the treatment effect defined step-wide. I go to detail the different steps. Step 1 corresponds to the definition of estimate. An estimate is a precise description of a treatment effect reflecting a clinical question. So first, we need to ask a causal clinical question. Then, we have to specify the protocol of the target trial. This means that we need to define the key component of the randomized trials that we are trying to emulate. The estimate should inform study design and analysis under different attributes: target population, treatment strategies, endpoint, intercurrent events, censoring rules, and finally, population-level summary of the treatment effect measured against some valid comparators. The second step is the selection of the external control data. To avoid the risk of selection bias, the closeness of the external population with the targeted population should be first required. External controls should use predefined eligibility criteria for the inclusion of studies. These criteria need to be defined to ensure patient similarity, relevant endpoint, and pertinent comparators. External controls will be directly selected from active arms from previously completed randomized clinical trials or reconstituted from real-world data. And finally, the last step corresponds to the indirect comparison of single-arm trial with the external control should be performed using appropriate statistical methods. Methods differ according to the type of data. External control arm may be established using both individual and aggregated data. If individual data are available for both groups, the commonly used method to mimic randomization is propensity score. Other approaches could also be considered, such as J-computation, double robust, or augmented IPW estimators. To address aggregated control data, population-adjusted indirect comparison have been proposed in the literature. The two most popular methods are matching adjusted indirect comparison and simulated treatment comparison. Once the relative treatment effect has been estimated, quantification of bias and robustness assessment need to be conducted. Unlike arm in randomized control trials, external control cannot control for unusual and non-confounders. Quantitative bias analysis investigates robustness of the findings under varying assumptions relative to a hypothetical unmeasured confounder.
Thank you. As you mentioned, there are several methods, statistical methods, to recreate the effect of randomization to get similar groups on their potential unidentified and measured confounding characteristics. And propensity score is one of these techniques. And then we can apply some matching to build pairs of similar patients, or adjustments to remove the effect of covariates, or the weighting to put more or less weight on each patient so that on average they are comparable, and also stratification to cut into similar subgroups. After applying one of these methods or several using sensitivity analysis, it's important to confirm that the methods permits that the groups are comparable. So we can use this kind of love plot, standardized mean differences for each variable, and to see how the methods that all the variables are now comparable after adjustment, matching, or weighting. So to conclude, during this presentation, we tried to show that RWE has a place in HTA and can address inquiries related to the target population and the real-world utilization of therapeutic strategies. In particular, external control arms may decrease the uncertainty of single-arm trial. But indirect comparison requires a complex and rigorous implementations to be valid, and recent guidelines from HTA bodies have started to clarify expectation.
This process would include a consideration of data quality, data comprehensiveness and completeness, and comparability to a potential experimental arm. If there are major issues with the validity or accuracy of the data, statistical methods cannot salvage the comparison. However, there exists a high-quality database referenced by the HAS. 2 weeks ago, the French National Authority for Health has referenced the ESME program breast cancer and lung cancer databases. Efforts are needed in the interpretation of the study with an external control. Bias, confounding bias, selection bias, survival bias cannot be totally eliminated. Study design elements can help to minimize bias, and statistical method may address the influence of bias on the estimation of treatment effect. Development of an appropriation of additional method and bias classifications are needed to appropriately characterize the offered data as a substantial evidence in the regulatory approval.
Thank you very much for your attention, and we are open to answer to your questions.
Yes, thank you, Thomas and Louise, for this interesting presentation. So do not hesitate to write your questions on the chat. And yes, there is a first question. Do you consider early access program as a real-world data or a clinical data?
Do you want to answer, Thomas? Or?
Yes. Early access program is at the frontier between real-world data and clinical trials, because patients are treated in the indications, and there is only follow-up using a specific follow-up, but it's at the frontiers, I think.
Like pragmatic trials that can be considered as clinical trial because it's randomized, but because it's very representative to the real world and the use, it can be considered also RWE. And it is considered in RWE by the FDA.
You may ask your question in French if you want.
Yeah, of course. And you can also speak if you want to tell me, just I will activate the microphone. So.
No question? We lose everybody, I think.
CPP and patient association. Super, we have some questions. Sorry. Do you want to ask it, Daniela, maybe?
Yeah, if you want. Are CPP and patients associated are trained to this new perspective? Is the first question.
That's a good point. And this kind of webinar maybe is a well way for the CPP, so ethical committees and patient associations, to be more aware of this kind of perspectives. But all the HTA bodies are working on the subjects and frequently publishing papers and communication. So it's going to change rapidly, I think.
Okay, thanks, Louise. And there is another question. Do you consider you would have any support for agencies to deal with these complex methodologies?
You want to answer, Thomas, maybe?
Yes, if you want. I think it's necessary there is a specific discussion between agencies and industrial before launching the external control arms.
The FDA have implemented some complex and innovative design, for example, for Bayesian design clinical trial, to discuss early in the design of the trial with the pharmaceutical company, and this kind of use of RWD could maybe be included in this kind of early discussions.
Yes, thank you. And another question. Do you think that acceptability by HTA bodies needs more cases or more implementation and technical knowledge?
Absolutely. It's quite new with a small number of use cases, for example, in France. But it's going to be more and more used and more understood by everyone, including HTA bodies. So, and HTA bodies are going to clarify also their expectations.
Yes, so the last publication by the HIS is an opportunity to deal with external control arm.
Okay, so another question. Scientific advices could be used, but often they are not enough pragmatic to provide complete recommendation. How could we improve this in your mind?
What is possible is some working groups with the scientific and also pharmaceutical company and also HTA bodies working together to understand what can be feasible, and but it's a work that needs to be implemented. That's a good point, Emmanuel.
Yes, very good point.
And speaking of complex methodologies, how do you imagine the integration of simulation data, mechanistic disease models, and real clinical data in dossier for regulation?
This point was not included in our presentations. We focus on real-world data with real data, but absolutely, this is an alternative that must be studied. Personally, I can't answer to this question because I'm not specialized in simulated trial.
Yes, I think this may be a complementary tool in a submission dossier for regulators.
In the future.
Yeah.
So another question, do you think that simulation and modeling could be used in close connection with real-world data to provide better evidence?
This question is quite close to the previous one, absolutely. Simulations and modeling should be based on clinical data and RWD to well predict what is going to happen in the real life after. So to provide better evidence. But that's a very interesting topic that needs more time to be discussed.
Okay, and Thomas, you want to say?
No, there is another question, I think.
Yeah. Is there an adjustment method recommended by the HTA bodies?
To my knowledge, there is no recommended approach, but the estimate need to be in the average target treatment.
The adjustment method really depends on the availability of data, the questions, so the estimate, as mentioned by Thomas. Several are cited by the guidelines, like propensity score, decomputations, doubly robust if you have individual patients' data, but there is today no one perfect solution.
Okay, and I have also a last question that what is the level of the use of hybrid clinical trial currently?
What is the use of hybrid clinical trial currently?
Yeah.
Very rare for the moment.
Yeah.
Okay, and what do you think about the risk of selection bias? How to control it or verify it?
Thomas, maybe it's in link with the estimate and the emulated trial can be a solution to avoid this kind of selection bias. And also external validity in any case, clinical trial, RWD, RWE, external validity must be assessed, and so sensitivity analysis and can answer to the business of several choices during selection analysis, and external validity through comparison with other data sources and other results is a way to see if, to verify if the population is not over-selected, for instance, or not specific to the question.
So yes, other questions?
Thank you for your very interesting questions.
Yeah, thank you very much.
Yeah, I have a lot of questions. Okay, so we wait a few minutes if you have some question, or otherwise we can go. Okay. So yes, thank you for your participation and for the speaker, of course. And we plan to do another webinar in April. So we will, the next topics. And thank you again, and see you soon.
Thank you very much.
Thank you very much. Bye-bye.
Thank you. Bye.